Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome to the Deep Dive, the shortcut to being well-informed, where we dissect the most important research to get you straight to the insight. Today, we're digging into a really massive, potentially transformative paper from Cell. It might just completely flip the script on how we view Alzheimer's disease. So, you know the standard picture, right? Sticky proteins, amyloid plaques, tau tangles building up in the brain.
The classic view, yeah.
Well, this paper says, hold on. The really shocking, kind of counterintuitive idea here is that Alzheimer's isn't fundamentally about those clumps. It's actually a huge systems-level communication breakdown.
Mm-hmm.
And get this, it seems to be driven mainly by the brain's support cells, the glia, acting almost like, well, saboteurs.
That really is the course shift they're proposing. For decades, almost all the therapeutic effort, billions of dollars, went into targeting amyloid.
The amyloid hypothesis, clear the plaque, fix the brain.
Exactly. The strategy was clear it out, cure the patient. But, well, we've seen trial after trial fail, unfortunately. Yeah. We got pretty good at cleaning up the gunk, you could say, in postmortem brains. But the cognitive decline in living patients often didn't change much.
Which forces you to ask.
It forces a really critical question. What if the plaques aren't the root cause? What if they're just a symptom, or maybe, just one piece of a much bigger, more orchestrated failure?
So researchers hit this therapeutic wall, and they started thinking differently about the brain. Less like a clogged drain, more like a whole city malfunctioning.
That's a good analogy. And it led them to look much more closely at the often overlooked partners,
the
glial cells.
Astrocytes and microglia.
Yep. The brain's infrastructure, its maintenance crew, its immune system, all rolled into one. Recent human studies were already hinting pretty strongly that the problem wasn't just in the neurons. It looked like a problem with how neurons and glia talk to each other, the neuron-glia crosstalk. These support cells, well, maybe they weren't supporting anymore. Maybe they were actively disrupting communication.
Okay, so if the problem is bad communication, just saying glias are involved isn't enough, is it?
Not even close.
So the mission for this paper must have been pretty ambitious.
Hugely ambitious. The goal was to build a really definitive, high-resolution map of the whole system specifically. The proteome, all the proteins, to pinpoint exactly which communication pathways were vulnerable.
And find the real control hubs you could actually target.
Precisely. It's a very deliberate effort to reframe AD as a network disorder. Move beyond that simple plumbing problem idea.
Makes sense. But mapping something that complex, I mean, the brain's proteome, that requires some serious methodological innovation, right? How do you even start?
It does, serious muscle. If you just look at a few hundred proteins, you miss the bigger picture, the coordinated network effects.
Okay, let's unpack that methodology because, honestly, tackling the sheer scale and complexity here is probably the biggest hurdle for anyone trying to really get this study. How do they handle all that data?
They use a really smart kind of dual mechanism. First, multi-scale proteomics. Yeah, meaning they didn't just look at one tiny bit of the brain. They profiled thousands, literally thousands of proteins across multiple vulnerable regions. Specifically, the parahippocampal gyrus, the PHG, critical, and the prefrontal cortex, the PFC. And importantly, they did this using multiple massive human patient groups, like the MSBB and ARSMAP cohorts.
Wait, why use several different big groups of people? Wouldn't one really good data set be cleaner?
It's all about statistical power and, crucially, reliability. If you find the same patterns, the same coordinated protein changes across different large populations, we're talking thousands of individuals here, you gain enormous confidence that what you're seeing is a real biological signal, however faint it might be, and not just some random noise or artifact specific to one group.
Okay, so step one, massive high-powered data collection from multiple angles. Step two must be turning all that data into an actual usable map of the disease. That's the network modeling part.
Exactly right. They used co-expression networks first, that groups together proteins that tend to rise or fall together. They behave similarly.
Okay.
But then, and this is really key, they applied Bayesian causal networks.
Causal networks, meaning they're trying to figure out what causes what, not just what happens at the same time.
Precisely. And they did this by integrating genetic risk data, like APOE status, directly into the protein network models. This lets them infer causality, or at least likely causality, not just correlation.
And that difference, correlation versus causality, that's everything if you're trying to develop a drug, isn't it?
It's absolutely critical. Correlation just tells you, say, protein A and protein B are both messed up in disease.
Right.
Causality suggests protein A might be the upstream switch that makes protein B get messed up, along with maybe C, D, and E.
So if you treat the correlative one, you might just be mopping the floor while the sink overflows.
Perfect analogy. But if you can successfully target a true causal driver protein, you have the potential to shut down the whole pathological cascade downstream. So this whole multi-pronged approach lets them find targets that are predicted to actually explain clinical progression, maybe much better than just counting plaques.
Got it. They're hunting for the molecular ringleaders,
Yeah.
the ones pulling the strings. So, okay, they built this incredibly complex map. What did it actually show them? What was changing in the protein landscape of severe AD brains?
Well, the changes were massive, especially in that memory region, the PHG. They found over 1,000 proteins that were differentially expressed, meaning significantly changed between the severe AD patients and the healthy controls.
1,000 proteins. Out of how many?
They analyzed around 10,000, so that's roughly 10%. 1/10 of the protein machinery they could measure was significantly altered.
Wow. That really shows the scale of the disease at a molecular level. Were these all proteins involved in neuron death, like you might expect?
Okay, here's the really striking part, the key takeaway from this section. They found that over 70% of these 1,000-plus changing proteins were actually upregulated in the AD brains. They went up, not down.
70% went up. That seems backwards, doesn't it?
It does, counterintuitive. When they looked at function, the proteins that were downregulated, the ones that decreased, they were almost entirely related to neuronal functions, synapses, communication.
Okay, so neurons are losing function, going quiet.
Exactly. But conversely, the huge number of proteins that were upregulated, they were heavily enriched for things like immune response, inflammation. And critically, the analysis showed these upregulated proteins were coming primarily from microglia and astrocytes, the glial cells.
So it's not just passive neuron decline. It's an active, almost aggressive inflammatory signal from the glia that seems to be overwhelming the system.
That's what the data strongly suggests, an active glial pathology.
Speaking of things being aggressive, we have to touch on the APOE4 gene, the biggest genetic risk factor. Did they see how this hyperinflammatory environment connected with APOE4?
They did. They looked specifically at one particular astrocytic protein that popped out called AHNAK.
AHNAK, okay.
And they tracked its expression levels across different APOE genotypes. What they found was really telling. AHNAK levels went up progressively.
How so?
Well, AHNAK expression was higher in AD patients without APOE4 compared to healthy controls, as you might expect, but it went significantly higher, really jumped, in AD patients who carried the high-risk APOE4 allele.
Wow. So AHNAK seems to sit right at that intersection of general AD pathology and the specific risk from APOE4.
It really suggests AHNAK isn't just some bystander protein changing randomly. It looks like it could be a critical link, maybe mediating or even worsening the disease progression driven by APOE4.
Okay, this is fascinating. Now let's pivot from the individual protein changes to the network failure itself. They have all this data syncing neurons, hyperactive glia.
Yeah.
When they plugged it all into their network models, what did the structure of that communication breakdown actually look like?
They found this core pathological structure, a subnetwork, that they labeled Module M3.
Module M3.
Yeah, think of M3 as the signature of the meltdown. It's a tightly connected group of proteins that beautifully captures this pathological three-way interaction. You see downregulated neuronal proteins directly interacting with activated astrocytic proteins and activated microglial proteins, all within the same module.
So it's not just one cell type going rogue, it's the whole local ecosystem failing together, reinforcing each other's dysfunction.
Exactly. It's the breakdown of the entire intercellular conversation. M3 really visualizes that network disorder concept.
Okay, M3 proves the network hypothesis. But still, you have this complex module, this chaos. How do you go from that to picking out a single credible therapeutic target like AHNAK? Why trust the model's pick over, say, proteins we've known about for ages?
Ah, that's where the power of the Bayesian causal network comes in. It wasn't just about finding proteins that changed. It calculated which proteins were the likely key driver proteins, or KDPs.
Key drivers, meaning?
Meaning these aren't just proteins that are abundant or change a lot. KDPs are the regulatory hubs. They sit in influential positions in the network, and the model predicts that if you tweak them up or down, they'll significantly influence the behavior of many other proteins downstream. They're like the critical switches.
So KDPs aren't the victims of the chaos. The model suggests they're actually the architects of it.
That's the idea. They identified 580 of these potential KDPs, and ranking very, very highly among them because of its high predicted causal influence, its origin in astrocytes, and that striking connection to APO4 we just talked about was AHNA.
I see. So all those lines of evidence pointed to AHNA as a prime suspect.
Exactly. It gave them the confidence to say, okay, this isn't just a correlation, this looks like a driver. Let's bet on this one and take it forward for definitive functional testing.
Which brings us, to the crucial validation stage, experimental proof. They picked AHNA, an astrocyte protein, and claimed targeting it could actually protect neurons. That's a big claim.
Yeah.
How do they set up an experiment to test that intercellular communication fix?
They used a really sophisticated, humanized co-culture system designed to be as clinically relevant as possible in a dish.
Okay, break that down.
Right. They took human astrocytes derived from induced pluripotent stem cells, iPSCs, specifically from individuals carrying the high-risk APOE44 genotype.
So high-risk human glial cells.
Exactly. And they paired these high-risk human glia with primary cortical neurons taken from a severe mouse model of AD, the 5xFAD mice, which develop plaques and tangles very aggressively.
So wait, high-risk human support cells talking to sick mouse neurons. Why mix human and mouse like that?
It's a clever way to isolate the specific interaction they want to study. You've got the human genetic risk factor embodied in the APOE4 astrocytes, and you've got highly vulnerable, pathology-prone neurons from the 5xFAD mouse as the target. This setup lets them ask, are the human glial cells, driven by APOE4, directly causing harm to susceptible neurons via a mechanism involving AHNAK?
Okay, it isolates that specific glial contribution.
Ah!
Makes sense. So the moment of truth. What happened when they dialed down AHNAK expression? Did a knockdown in those high-risk human astrocytes? Did the neurons actually get better?
The results were, frankly, striking. The protective effect was pretty immediate and clear. They measured the neurons' electrical activity, their ability to fire and communicate. Neurons that were grown alongside the astrocytes where AHNAK had been knocked down showed a significant 1.8-fold enhancement in neuronal spikes.
1.8-fold. What does that mean in practical terms? Is that a tiny change or?
Yeah. Oh, that's highly significant. In these kinds of functional neuroscience assays, a 1.8-fold increase isn't subtle at all. It's like taking a neuron that's really struggling to fire and bringing its activity level way back up towards a healthy baseline.
Wow. So they rescue the electrical communication just by tweaking a protein in the neighboring support cell.
Precisely. And it wasn't just electrical function either. They also looked at the molecular hallmarks of degeneration in the culture. They saw a reduction in phosphorylated tau levels, pa-tau, and even some modest reduction in A-beta levels as well.
So signs of reversing the actual degeneration too.
Yes, demonstrating a rescue of key neurodegenerative features.
Incredible. So, okay, let's zoom back out. We started this deep dive with the old idea of AD as just clogged pipes, basically. Yeah. And we've ended up with compelling evidence that it's this complex, orchestrated network breakdown driven significantly by glial cells and potentially fixable by targeting specific communication hubs like AHNAI. What are the really profound translational implications here? Where does this take the field?
Well, the huge innovation here is moving beyond just description or correlation. We now have a map driven by predicted causality, validated experimentally.
Right.
And that means the field can potentially pivot. I mean, really pivot. Instead of pouring all resources into trying to, as you said, cure the lesion, clear the plaque, we can now focus effort on trying to repair the network. It fundamentally shifts the therapeutic strategy towards modulating how these cells talk to each other.
And presumably this study gives us a list of potential targets for repairing that network.
It absolutely does. That's maybe the biggest treasure on Earth here. Those 580 key driver proteins they identified. That list represents a foundational blueprint for next-generation AD therapies.
580 potential targets.
And here's the kicker, maybe the most astounding part for me. They checked how much these KDPs had already been studied in AD. A massive 72% of them, that's 340 distinct proteins, were found to be seriously understudied.
Understudied.
Meaning they appeared in fewer than 5 previous Alzheimer's research papers. The field, by focusing so heavily on amyloid and tau, was potentially missing this huge untapped continent of other critical players and potential targets.
Incredible. So we went from thinking AD is about one or two bad proteins to seeing it as this vast interconnected network failure, specifically at that crucial glia-neuron interface. And proteins like the astrocytes AHNA are emerging not just as markers, but as potential master regulators, actual targets.
Exactly. Which leads us to maybe a final provocative thought for you, the listener, to consider.
Okay.
If this kind of deep proteomic network analysis applied to Alzheimer's can reveal this level of hidden complexity, pinpoint hundreds of new understudied causal targets, well, what percentage of other complex chronic diseases might be driven by similar, currently undiagnosed network failures?
You mean like Parkinson's or ALS?
Parkinson's, ALS, maybe even things like type 2 diabetes or certain autoimmune disorders. Could they also be fundamentally network problems that we've been missing because we weren't looking with the right tools? Could we unlock treatments for those by applying the same systems-level, network-first approach? The true revolution here might actually be the methodology itself, showing us a new way to look at disease.